human adult body is composed of hundreds of thousands of billions of cells. So huge amount of cells are developed from a single cell, the fertilized oocyte or the zygote. So imagine if there are any genetic or epigenetic abnormalities happens in this single cell zygote, later probably all these cells in adult body will be affected. So my laboratory trying to using the combination of several different approaches to analyze the gene expression regulation in human early embryos. So the first one we're trying to using the single cell RNA-seq approach we developed in 2009 to see how the gene expression dynamics in these human early embryos. And after that, we want to go to upstream to see how the genetic and epigenetic regulation it is. For the genetic regulation, we collaborated with Professor Santé's lab to using the single-cell genome sequencing technology they developed to see how these genetic polymorphisms in these human embryos contribute to the gene expression regulation. And for the epigenetic aspect, we're trying to develop some low number of cells or even single-cell epigenome sequencing technology to see how the epigenetic layers contribute to the gene expression. And after we find some interesting candidates, we can go back to the mouse in vivo development model or the human ESL differentiation in vitro model to see how they regulate the gene expression. So several years ago, we collaborated with Professor Recherche's lab at Third Hospital of Peking University to see how the pre-implantation embryos there is gene expression dynamics. We analyzed from the mature oocyte until the blastocyst stage using a single-cell RNA-seq approach. So we find the general gene expression dynamics. For example, if you're looking at this unsupervised hierarchical cluster analysis, so the gene expression in these pre-implantation human embryos, they separate before 4-cell stage and after 8-cell stage. So before 4-cell stage, as you know, is mainly maternally controlled, and after 8-cell stage is mainly the zygotic expression. So after we find these gene expression patterns in human embryos, then we want to see how it's genetic and epigenetic regulation. So for the epigenetic regulation, one of the probably most well studied is about the DNA methylation. And usually the DNA methylation is very stable, but during the early embryonic development, there's two waves of global demethylation and remethylation. The first wave is after implantation. There's a global demethylation, and the lowest methylation achieved as a blastocyst stage. And then after implantation, it remethylated, and in the somatic cells, it maintains this hypermethylation state. But in the germline, the primordial germ cells, it will be demethylated again and remethylated differently in the sperm and oocyte. So this is relatively known in mouse, but not too much known in human. So because the technical limitation, so we try to develop the single-cell DNA methylome sequencing technology called single-cell RBS. So the method is in fact quite straightforward. So the standard RBS technology can use hundreds of cells to analyze the DNA methylome. But the main problem is there's 6 purification steps in the protocol. So we're trying to make them into a one-tube reaction to make all the buffers compatible with each other. And then after every step reaction, we just heat inactivate the enzyme, add the enzyme for next step. So in this way, we make the work for a single cell. From a single mouse embryonic stem cell, we can analyze about 1 million CPG sites methylation pattern. It's about 40% compared to when you're using hundreds of cells. So using this, we analyze these human early embryos from the sperm oocyte until the post-implantation. And if you're looking at global methylation pattern, it's very similar to mouse. First global demethylation, and later after implantation, remethylation. But one of the major differences is here. So in mouse, the major demethylation finishes at late 1-cell zygote stage. But in human, this is delayed. There's a lot more demethylation from the 1-cell to 2-cell stage. And if you're looking at the methylation on the gene body, it's similar to in the somatic cells. There's a hypomethylation valley around the transcription site and hypermethylated on the gene body. During this first global wave of demethylation and remethylation, the imprinting genes, gel methylation, are not affected, so they are accurately maintained. So here is an example. This maternal imprinting gene, KCNQ1, you can see it's fully methylated in the oocyte, no methylation in sperm, and after fertilization, it maintained 50% methylation in all these state embryos. So meaning they maintain their parental specific methylation. And more than these imprinting genes, people know there are also a lot of other oocyte or sperm-specific differentially methylated regions. But where are they? So because we do this whole genome screen, so we can really see where these DMR regions are. So using the 100 base pair tiles as a unit, so we find more than 12,000 oocyte-specific DMRs and more than 17,000 sperm-specific DMRs. And their methylation pattern is very different. For example, for the oocyte-specific DMRs, it's fully methylated in the oocyte, no methylation in sperm, but after fertilization, it maintains this about 50% methylation, meaning that they can transiently maintain this imprinting state. But different from the standard imprinting gene, so after implantation, the unmethylated paternal allele is also fully methylated, so meaning they lose the imprinting state after implantation. And if you're looking at the sperm-specific DMRs, it's very different. So it's fully methylated in sperm, no methylation in oocyte, but after implantation, the methylation is quickly disappear. So mean even for these sperm-specific DMRs, they can even not maintain the transient imprinting. But after implantation, both alleles are remethylated, become similar to the oocyte-specific DMR regions. So we find so many new DMR regions, so we want to know if there are any functional significance. So we analyze their localization in these functional human genome elements. So we find the left side is about these oocyte-specific DMRs. So the height of this heat map is proportional to the number of the 100 base pair tiles. So you can see these oocyte-specific DMRs, they're strongly enriched on the CPG islands in the gene promoters, whereas the sperm-specific DMRs, they strongly enriched on the somatic cell-specific enhancers. So we think probably for these oocyte-specific DMRs, they are methylated specifically in oocyte, a large set of these CPG islands are promoters to repress these target genes expression. But for these sperm-specific DMRs, they methylated a large set of these somatic cell-specific enhancers to repress these somatic cell-specific genes abnormal expression in the male germ cells. As we mentioned, that there is global demethylation from sperm and oocyte in the zygote, but we know that the sperm methylation is much higher than oocyte. So how about these two maternal and paternal genome demethylation dynamics? Are they similar or different? So here we really need to separate paternal and maternal genome by the microcapillary, and then we can do the half-cell demethylome sequencing to see accurately how the paternal and maternal genome demethylation dynamics in the same zygote. The problem is for these human embryos, the paternal and maternal genome, or the male and female pedunculae, they are very difficult to discriminate. So first, from every zygote, we just separate the two pedunculae, and then we do the methylation sequencing. And then because we identified a lot of oocyte or the sperm-specific DMR region, so we can base on such region to determine which pedunculae from the zygote is male, which one is the female. And for example, for this first zygote, the two pedunculae, you can see the first one, the demethylation pattern very similar to the sperm one, the DMR region, and the second one very similar to the oocyte. So we know the first pedunculae should be a female pedunculae, the second should be a male one. Sorry, the first is the male one, the second is the female one. So in this way, we analyze different zygotes because for these zygotes, we do the ICSI, the intracytoplasmic sperm injection, so we can accurately know the fertilization time. So you can see there's 2 points here. The first is after the same time of after fertilization, the different per nuclei, their methylation level is very different, very heterogeneous. So that means in different embryos, the demethylation speed is very different. Some are faster, some are slower. And the other point is that the global demethylation in sperm is or the male per nuclei is much faster than in the female per nuclei. So just at the late zygote stage, the methylation from the father is already lower than from the mother. So previously, people believe that for these mammalian embryos, for these maternal genome, is mainly demethylation mainly happens through the passive demethylation, and the paternal genome demethylated mainly through the active demethylation, and for the active demethylation, usually it goes through the hydroxymethylation through the oxidation by the tight proteins. So we do the staining in these human embryos, and very unexpectedly, we find essentially for every embryo, both the male and female per nuclei have a lot of hydroxymethylation, so indicating that even for the female per nuclei or the maternal genome, there's also a lot active demethylation. So because we cannot do any genetic manipulation of human embryos, so we go back to the mouse system to collaborate with Guo Liangxu's lab and Jin Li's lab to using their mouse model to see how this active demethylation of the maternal genome. So we find, in fact, a lot of genome regions are actively demethylated in these maternal genome. For example, here is an example. You can see this gene locus JBTP32. It's fully methylated in the oocyte, strongly demethylated in the maternal genome or the female per nuclei. But when you knock out TET3 to block the active demethylation pathway, it cannot be demethylated anymore. So meaning that it's definitely active demethylated region in the maternal genome. And if it's actively demethylated, usually the two strands of the DNA should be both demethylated. So we can see here is a case, for example, for this gene locus, you can see in oocyte is fully methylated, and in the wild-type female per nuclei, some of these DNA molecules, both strands are fully unmethylated, so indicating that it is really actively demethylated. And we saw that this active demethylation finish, it goes through the hydroxymethylation to FC and CAC and finally becomes the unmodified cytosine. Usually this final step should be mediated by the TDZ pathway. But when we do the TDZ knockout, very unexpectedly we find the demethylation still completed, but it still it don't depend on the TDZ pathway. So it must have some parallel pathway to finish this final step of the active demethylation in these mammalian zygotes. So people already developed a lot of different single-cell RNA-seq approach, such as what we developed in 2009. But the problem is all these known single-cell RNA-seq approaches can only analyze messenger RNAs with poly tail. So if it's poly minus RNAs, nobody can analyze them in the single cell. So we recently developed a new single cell approach called SuperSeq. It can analyze both the poly plus and the poly minus RNAs. So the idea is straightforward. So if we're using a random primer to do the reverse transcription, then we can also recover the poly minus RNAs. So we saw the method is quite reliable because we pulled 10 HEC293 together, and then we lysis them and then split into 10 different tubes to create 10 average single cell, and we saw that their transcriptome looks very similar, so meaning the technical variation is minimal. And in this way, we can detect a lot of poly minus RNAs. For example, the circular RNAs recently found in the mammalian genome, and they usually probably have a lot of important functions. So we are using our single cell new SuperSeq approach to analyze these mouse early embryos, and we find about nearly 3,000 circular RNAs expressed in these mouse pre-implantation embryos. And we can also analyze the total amount of messenger RNAs in the mouse early embryos. The first is more than 20 million in these mouse oocyte, in a single oocyte, and later strongly decreased, but later it come back. And if you're looking at these circular RNAs, so these maternal ones are quite resistant to the maternal degradation, and later there's also some zygotic ones. And here is circular RNA abundance compared to their linear host genes. Usually it's between 1 to 10%. And the absolute copy number is about 10 to 100 copies in every single embryo. So more than these circular RNAs, we can also detect a lot of other polyamorphous new mRNAs. For example, these one you can clearly see is maternal ones is strongly expressed in the oocyte and zygote and later decrease. We also find some later zygotic new polyamorphous linear RNAs. And just based on these new linear RNAs, if we do the PCA analysis, you can see these mouse pre-implanted embryos can clearly be separated into different states at a very ordered manner. And after we analyze these human mouse pre-implanted embryos to make sure the technique works, then we go back to the mouse human pre-implanted embryos to see how the polyamorphous RNA expression there. And you can see here is the absolute copy number in each human embryo. For example, in a human oocyte, the total messenger RNA copy number is about more than 50 million copies, and it's gradually decreased. The lowest is achieved between 4 and 8 cell states, and later after the zygotic activation, it recovers, and when it go to the late blastocyst states in one embryo, and there's more than 100 million messenger RNAs. And using this new method, we can also see these circular RNAs, although they don't have the polytail. So we find about 10,000 new circular RNAs. They are derived from about 3,000 host genes, and the majority of them are with several exons, and quite a lot of them show developmental stage specific expression patterns. And if you compare between the mouse and human, these circular RNAs, and you find a lot of them are conservative, and they mainly related to the chromosome organization or the cell cycle. So more than these circular RNAs, we can also find these linear polyamorphous RNAs in these human pre-implanted embryos. For example, we can find a lot of these maternal non-poly linear RNAs. They mainly express in the oocyte zygote or before the 4 cell stage. And we can also find some zygotic specific new polyamorphous RNAs. And we, 2 years ago, we collaborated with Professor Santé's lab to analyze these human embryos at the zygote stage. We can analyze the first and second polar body, and then to predict the nucleoid situation in the embryo. And we saw that the method worked. For example, if you analyze the first and second polar body, you can deduce what's the nucleoid in these female prenucleus. And when we really isolate these female prenucleus and sequence, we can verify what we predicted. And we in total analyzed 8 donors, and we find different people have different percentage of these nucleoid oocytes. And on average, the nucleoid situation is about 17% in all these 8 donors together. We can also using this method as 8 cell states, embryo states, to see how the aneuploidy situation. You can see, for example, for this first embryo from the 1 cell, you can clearly see the gain or loss of the chromosomes. But the problem is we can only for one cell, we either analyze their genome, then we don't know their transcriptome, or we analyze their DNA methylome, then we don't know the genome or transcriptome. So we think maybe we need to combine from the same single cell. We can analyze both the DNA methylome and the genome, and then we can know the relation between them. For example, if the embryo have aneuploidy, do their DNA methylome also affected or not? So we recently do some new work to analyze these single cells for these human pre-implanted embryos. So we do the DNA methylome sequencing, but at the same time, because when you do the DNA methylome sequencing, or you also sequence these DNA fragments, and then you can count these DNA fragments to get the copy number variation information. And then we have a chance to see if this aneuploidy will affect the DNA methylation or not. So now we in total analyzed about 131 single cells for these human pre-implanted embryos. We do the DNA methylome sequencing, but at the same time we can also see the copy number variation of the genome. And you can see in total, in general, a lot of these embryos show aneuploidy. For example, for example, for these metaphase 2 oocyte, we find about 11% have these aneuploidy situation. And if we count the aneuploidy in all these embryos together on each individual chromosome, we find it seems enriched on these chromosome 18 and chromosome 21 abnormalities as expected. So here are some examples. For example, when we do the DNA methylome sequencing, we also see the copy number variation. This first oocyte is totally normal, but the second one you can see it lost a copy of the chromosome 10. And for this 2-cell embryo, you can see for this first embryo, the 2 cells both are normal, diploid. But for the second 2-cell embryo, you can see for this chromosome 8, it lose one fragment from both of these 2 cells. So it means it must be from the zygote. And for the 4-cell stage embryo, you can see the first embryo, the 4 cells are totally normal, diploid. And for the second 4-cell embryo, you can see it lose one copy of the chromosome 5 in one blasomere, and it gain a copy in the other blasomere, but normal in the other 2 cells. So that means it should be a mitotic aneuploidy. So during the cell division, one cell gain one extra copy of a chromosome, the other just lost one. And if we're looking at the trophic cells in the blastocyst stage, and is similar. Some embryos are normal diploid, some cells are become aneuploidy. And the more interesting thing is the connection between the aneuploidy and the DNA methylation. So our preliminary results show that if the cell gain extra copy of the chromosome, then usually the chromosome will have more chance to become hypomethylated. And if the chromosome copy number is lost from the 2 copy become 1 copy, then this chromosome will tend to be either hypermethylated or hypomethylated. But we still need to accumulate more data to verify this. So it seems to us these aneuploidy not directly affect the embryo genetics, but it also indirectly affect the DNA methylation changes. So previously, what we saw in this human pre-implantation, the DNA methylation, is we using the single-cell RBS technology. So for this one, we mainly focus on the CpG islands or the CpG enriched region. But if we want to see more on this CpG sparse region, so we need some new method. So we use the method recently developed by Wolfric Lab, and they developed the single-cell base-pair sequencing technology. So using this method, we analyzed these human pre-implanted embryos, and you can see what the pattern we find in general is similar to what we found using the single-cell RBS. You can see the hypermethylation in sperm, hypomethylation in oocyte, and then get global demethylation until the later stage. And if we separate the different genomic regions, You can see, for example, for the CPG islands, in fact, in the sperm, the methylation on CPG is already lower in general than the oocyte. And later in the pronuclear states, you can see the CPG islands in the male pronuclei is much lower than the female ones. But if you're looking at these repeat elements, for example, for the lines, it's different. It's hypermethylated in sperm and hypomethylated in oocyte, and in the pronuclear states in the zygote, the methylation become comparable, and later it further decreased until the blastocyst stage. However, if we're looking at the enhancer regions, you can see it's strongly methylated in sperm, lowly methylated in oocyte, and later in the pronuclear states it become comparably methylated, and later it further globally demethylated. So finally, to summarize, we both for the pre-implantation and the post-implantation primordial germ cells I now mentioned here, we now can really have an accurate idea how about the demethylation dynamics there. So now we know in these human sperm, the global methylation level is about 86%, very high. And the oocyte is a little lower, 72%. And then during the pre-implantation development, is globally demethylated. And as the blastocyst stage, the methylation about half left, about 40% methylated. And later, when during the implantation, it's globally remethylated again. And in the early post-implantation embryo somatic cells, the methylation level is around 92%. And then it generally maintained in these somatic cells, including both ectoderm, mesoderm, or endoderm. But in the germline, the primordial germ cells, it globally demethylated again. And at around 10 to 11 weeks, in these human primordial germ cells, the methylation left is already only about 6 to 7%. And on this functional enhancer region is even less than 3%. So essentially, in these primordial germ cells, these functional genome elements are free of any demethylation. But interestingly, we also do some immunostaining. We find that because for these early human primordial germ cells, they lose the methylation nearly completely, but their gene expression don't change too much. So we guess probably there's histone modifications, other layers of epigenetic regulation is still maintained there. So we do some immunostaining, for example, for the F3K9 trimethylation, we see the heterochromatin mark. We can see the pattern is still the same in the 10 to 11 week PCs compared to the early PCs or the neighboring somatic cells. So we think during this PC development in vivo, the demethylation is first uncoupled from the gene transcription regulation, and then it's mainly the histone modification control the gene expression program and maintain it stable in these human germline. And later, when they mature, the germ cells mature, become oocyte or sperm, they are remethylated again. So the logic is first, during the pre-implantation development, because the sperm and oocyte have their own gamete-specific methylation pattern, these ones need to be removed and permit the embryo to set up their own methylation patterns. So the gamete-specific methylations are removed. But because there's other methylation similar between the gamete and the somatic cells, so they are just kept. So that's why in the pre-implantation, only about 50% of the methylation are lost. So another half just keeps. And then during the post-implantation, during implantation, the methylation is rebuilt. But because at this time, the whole embryos haven't differentiate into the germline and the non-germline. So the embryo remethylates the genome following the somatic cell pattern. And after the implantation, in these somatic cells, no matter mesoderm, endoderm, or ectoderm, they maintain this hypermethylation pattern. But in the germline, they need to remove these somatic cell-specific methylation to permit to remove all these all these epigenetic memory and permit the germ cells later to rebuild their own methylation pattern for the next generation. So for this second global demethylation is much stronger, from 92% to nearly nothing left. But still, if you're looking at these repeat elements, there's more methylation left there. So probably they offers some explanation for the transgenerational epigenetic inheritance. So finally, I want to thank my collaborator, Professor Sandy Xie, Jie Qiao, Rui Qiang, Guo Liang, and Qin Song. And my work is funded by the Chinese different agencies. Thank you.